Okay.
To a certain extent, contrast with it, on the right hand. Probably just turn it.
We just kind of talk to folks, isn't it? Yeah.
No.
Yes. Can we go first?
Yes, I just want to make sure. Cool.
Okay.
Wait. Do you mind looking for
All right. So my name's Carl Shefsik,
And I'm asking that.
and we were partners during our internship. And basically, to understand what our internship was, you have to understand how we got our internship. So we basically received our internship from one of our mentors and colleagues at school, Samantha Chavez. She basically came up to us and asked us if we wanted an internship with our mentor, Emma Borgeson. And we said yes, of course, it sounds amazing. We'd love to work in her lab, and yeah, from there we started working in December up until our internship here this month. And then we just hit the ground running from there.
So as Carl mentioned before, we got our start with a mentor 6 months ago. And then from there, right when we met her, we went through a whole interview process and that sort of thing, and then we went through different training. So we were trained on different lab management and different tasks we perform in the lab. So then when we started our internship, we could just start with the project right away. And so our overview for our presentation today, which we already covered our background, we're going to talk a little bit about lab management, our general project, what we did over our internship, and then a brief. So as I mentioned, we learned about general lab management. So these tasks are very helpful for when you're in a lab setting. Little things like washing dishware and learn how to autoclave, which is sterilizing dishware, so that goes a long way, as much as it might not seem like a lot, but it actually is. So we learned little stuff like changing water baths, benchtop paper, checking for hazardous supplies, CO2 tanks, that sort of thing, and then it made a big difference when we started working in the lab, because we kind of had a background knowledge of everything, or how everything works.
Right, and it helped us understand where everything was in the lab as well. It was very easy to identify and do things that we needed to do when we needed to do them.
So, like we mentioned, we're going to go through a general project overview. So our project this semester covered immunohistochemistry, imaging, tissue culturing, MS studies on typically obesity, so we'll cover a little bit about that. We did study quantification statistics and creating algorithms.
Right. So immunohistochemistry is a big field in which many things happen, such as the staining that we do for certain tissue and cells. These stains can range from H&E series red PAS, which...
Yeah, so H&E stands for hematoxylin eosin. The hematoxylin is typically like a dark blue purplish color, and it stains for the cytoplasm in your tissues. Then the E in H&E stands for eosin, which is typically a light pink color, which stands for the nucleus in cells. Then you have the series red staining, which obviously is a red stain. It stands for the collagen, which acts like the glue in the body. So typically in areas of inflammation, you'll see lots more collagen, or a lot more collagen. So that's what we stain for, we're kind of looking for. And lastly, we have the PAS stain, which stands for periodic acid shift stain, which comes out as like a bluish color that we stain our kidneys in, and our kidney samples in, and it stains for, or we're looking for the misandrogen cells. Another stain that isn't really mentioned up here, but we briefly kind of covered, was Mason trichrome, which the colors that come up from there are green, blue, red, brown, and black, and it stains for things from keratin to muscle fibers to nuclei to, like, there's a broad spectrum that it stains for.
Right, and these all are stained to different types of tissues and cells that we use. So in order to do this, we first put the tissue into a type of mounting wax or a mounting type medium. And then we use that, chop them up at 5 micrometers. 25
which is
micrometers.
around the length of the cell layer.
And then we put that onto a slide, which then gets transferred over, dehydrated, and stained whichever stain we want. and then rehydrated, and that's when we put on a cover slip using mountain medium. They can be anhydrous, and we use anhydrous, non-aqueous, which means they're hydrophobic, don't like water. So it's able to retain and hold the cells in, so you can keep looking at them over and over.
So as Carl mentioned, when the slides are put in, or the tissues are put into the paraffin wax, it kind of holds the cells together, and then, or holds the tissues and cells together, yes. And so then when it's brought through this process, first it's put into xylene baths, which strips the wax from the slide, and then it's rehydrated through 5-ethanol baths into 3 concentrates. And then finally through PBS, which stands for phosphorus saline, and then from there we're able to stain. And so that process takes, the actual going through the xylene and the ethanol baths takes about roughly 10 or 15 minutes. And then it's incubated, it's left in the stains for 2 hours, and then brought back through, rehydrated. And then we cover slip, as you mentioned, and then it's left over extra.
Imaging. So we take images of these cells afterwards, and then use those images and identify and analyze what we're trying to get out of them. So using the program's image scope and Infinity Analyze, we're able to do exactly that. So when we capture images on the microscope, we use Infinity Analyze. And, yes. We use Infinity Analyze to look at them and make sure we have the right pictures we want to take. So we can change the contrast, we can change the brightness, different types of gamma and gain to necessarily bring out the cells and have a clear image of what we're trying to see. So we are looking for, in our study, adipocytes. And so adipocytes are basically fat cells in a nutshell. So brown adipose tissue and white adipose tissues both have adipocytes, but they do different things.
So with white adipose tissue, the white adipose tissue, which is typically found on your body, like in your arms and legs and stuff like that, that stores fat and hormones. And then for brown adipose tissue, it's stored in the upper neck region and back and chest region, and it can just heat. So a lot of times newborns have higher levels of adipose tissue. So I thought it was interesting that they're known not to shiver, which I thought was interesting about that.
Right. And then we also analyzed for glomerulus because we wanted to find out different things about the kidneys, since obesity is generally in Emma's theory to chronic kidney disease.
So basically, the glomerulus is a structure of capillaries located before the nephron. The nephron is located inside the kidneys. And so the nephron's main function is to filter out your blood, and then any waste is then turned into urine. And so it goes to the nephron, and then the first part of the nephron is the glomerulus. And so it's kind of like a hose-like structure where it moves like this. And so it passes, or blood's filtered through there, and then passes through the rest of the nephron.
And then tissue culture. So this was another thing we did in our project. We learned how to do tissue culture. So tissue culturing is basically when you culture tissue outside of an organism's environment. So, such as petri dishes have acar that you can use to grow bacteria. We grew an immortalized cell line to basically learn how to do tissue culture and learn this technique. So
the protocol for tissue culturing is first you warm the media, and so the media that's placed into this glass right here is what the cells basically use to live on, and so it's kind of for food, the protein, that sort of thing. The media is composed of 3 different things, or the media stands for DMEM, which stands for glucose modified Evo medium, and so in there it's composed of L-glutamine, which is which is a protein. Then it's also composed of test with ES, which is
it's an antibiotic to keep it sterilized.
And then finally it's serum to kind of give it its color. And so then after we warm the media, then we are able to disinfect ourselves and go into the hood. We retrieve ourselves from an incubator, which acts like the body. It's a and it's kept at about 37 degrees Celsius, which is like our body's natural temperature. And then from there, we are able to then split the cells. We do this about 4 times. I remember 4. And so we do this by, as you see, Carlos' pictures, removing the dead cells. So,
so we remove the dead cells first. Then from that, we add some more serum. Media. Add some more media to this bottle. And we use that media to loosen up the cells that have been attached to the wall of the flask because they want to grow and create tissue. So we loosen up those cells, then we transfer those cells into another clean flask and with new media to keep them growing and living healthy. Right.
So our results from this were kind of interesting. I actually grew a bacteria colony in my cells, which is pretty interesting.
That big, it was
huge and kind of disgusting. But it was actually pretty cool to see as students kind of go through this whole process and see the result after that. Or generally when done correctly, you have cells that can go on.
So back to obesity. So what is obesity? Basically, obesity is when your body and your BMI is basically just over the average amount. It's overweight and it can be caused by many different things. Generally, it is caused by overeating and lack of exercise, but you can be genetically predisposed to obesity and other metabolic and hormonal functions can also contribute to obesity.
Obesity matters because it is proven to be fatal a lot of times, and so obviously it's also linked to other diseases.
Right, obesity can cause many different diseases. It's a high leading factor, and therapeutics and treatments can work internally and externally, such as just regular dieting and eating. Right, I said the same thing twice. And also internal, like I said, such as injections or pills can also help decrease your obesity. So it is linked to chronic kidney disease because obesity can cause inflammation over time in different areas of your body and effectively cause cells to not do what you're trying to do. And basically, it kind of destroys the cells internally and gets rid of their structures. And right, yes. So it negatively hits the kidneys. So.
And so part of this was we learned how to quantify adipocytes, which we mentioned for our fat cells. And so we did this with a new program called adipocyte quantification or application. And so first we analyzed the cells using a microscope, and then we brought them through image scope, which we used to circle these adipocytes, like each and every one of these we circled by hand.
Right, we do it manually.
And then we exported this data onto a spreadsheet to keep, that you have all the areas and stuff like that. But that takes a lot of time usually. So this new program that was developed was able to actually do the circling automatically and then export them which is neat.
But there's a drawback, seeing as the program was designed specifically for different types of staining. So to replicate exactly their types of cells and their types of stains and pictures would be very hard to do and didn't very cohere with our project already. So there were many problems, like in the next slide, where it wouldn't capture all the adipocytes that came up. We would only get a couple. It'd be kind of hard and shaky, but it did do its job sometimes. But for the most part, we stuck manually doing it because it was tried and tested.
So this was a good process for us to undergo because we were learning how to kind of picture general hypotheses, like will this work to quantify all of our images at once and export them simultaneously. So we pitched our hypothesis, so we did a few trial errors with different, we tried different, raising the brightness or contrast, something like that, to try and bring out more of the areas around the adipocytes. So we tried different techniques, and then finally we were able to come up with our results. And so the next we tried again, pitch different hypotheses. So it was a really good scientific process that we were able to undergo. and only a few show you.
Right. So, taking the data that we exported and from our manual circling, we then took these and put it into Graph at Prism. So this program is basically kind of like Excel, just like on steroids. And it helps us build graphs and analyze our data using different scientific tests such as the p-tests and yes, that. And so these string bar graphs show us the numbers and averages of the circles that we circled.
So basically, I mean, just like you would do in an Excel sheet, we inputted our data manually into a spreadsheet, and then if you kind of see that like bar on the left-hand side there, it then computes it into a graph, which is here, which you can label and then.
So right, so all these lines show the difference or the p-value of our test and of our data. So the more the stars, the higher the p-value, meaning, or the lower, actually. The lower the p-value, meaning that there was more difference. So it was better, in fact, more likely that your study was right. So.
That's basically how you. And so finally, we both did a little bit of analyzing these same tissues using algorithms. So basically what an algorithm is it's kind of like a template, if you will, that separates whatever you're trying to look for or identify in the program. So it's kind of a broad explanation. But the algorithm that we use is called color deconvolution, and version 9. And that program that algorithm, what it does, it separates each pixel count, into different colors. So, for example, sorry, I don't have an example for you, but your weak positive pixel count would be like counted as yellow, whereas your medium would be counted as orange, for example, and your strongest would be counted as red. So this is a quick way to be able to go in there and then see, like, oh, we're looking for a weak positive pixel count. We'll go this is the area that's all in yellow. And so when we were creating these algorithms, the algorithms were generally created so that we were able to tweak them a little bit and then apply a macro. And the macro is kind of like the template that goes on top of this template to then go in and fine tune what we're looking for. So for example, when we were looking in blue marrow line, we were like, well, we want to find the sangin, which acts as the the blue is kind of a rough definition for it. But so then we were able to say, okay, well, we want the misandrin to be yellow, whereas we want the nuclei to become blue. So that's what we did there. And so with these analyzed images, then we were able to take the data collected from them, so like for I said, the weak positive pixel count, and then plug it back into the graph baptism, learn our statistics, and multiply our graphs.
So this project basically was incredibly insightful. It was great to go to the lab and work there for a long period of time, since December. And yeah, we learned a lot from just tissue culture, like we said, to a whole study on obesity and chronic kidney disease.
One of my favorite things is like every day when I come to the lab, I learn something completely different. So I end up with this field with like just different key points that Emma would like mention to us. So I think that was one of my favorite things. And so like, later in life, I do see myself going into research, or if not research, then something still in the science field, in our biochemistry.
Right, yeah. I also see myself going to the field of biology, probably a biotechnician, but it was still fun and great doing medicinal chemistry as well. I really liked it. I was like, it's amazing.
Very good.
Yes. Yes. So. Now the floor is open for any questions. Oh yeah, those are just kind of
Any questions?
we've had a lot of you guys, but yeah.
Anything about internship or finding an internship?
So, you guys just get one classical and say, like, we have some action, you guys want to do it, or was it like a lot of work?
A lot of both. So
Yeah.
we were basically offered this great opportunity from our upperclassmen at our school, Samantha Chavez, like you said. And so she came to Jesse with this internship opportunity. And then me, Madison, and somebody else fought for this. And then ended up me and Madison kind of just battling it out. And then we kind of were just too good. And so they kept us both. And so
It was fun to go through like the interview process and that sort of thing, and then the training took forever. So, yeah.
But this is a good example of knowing early on what you want to do. Like, Jackie came to me and I remember October and said, oh, you know, Samantha was our intern last year, you know, she has connection. Who do you know any of our students that are interested in this sort of work? So that was a big boost for them that they had already identified this is the sort of internship that I want. Whereas if in September they had been like, I don't know, architecture, art, you know, they've been kind of wishy-washy, like our minds went to them instantly because we knew they'd express this strong interest.
And then
So that's another reason to kind of think about over the summer. You know, you can start junior year really knowing you're going to be in a better position.
And then not only that, but then Samantha also, she completed an internship last summer with Emma. So don't be restricted to just doing it in May. Be open to be doing it throughout the entire semester possibly, like maybe on Mondays and Fridays if we did, or maybe during the summer so you can start that connection now, and then when May comes around, you're like, oh hey, I already have one, and just keep going here sort of thing.
yeah, just getting early lead is your best.
Because a month is too short to do anything with students, especially at this age. So the fact that you guys have been in these 6 hours a week for 6 months is huge. Because then you can basically teach them everything. And then we've had special seminars where you guys have had a day off school, and then you've actually been in all day. And like we've done training, and then we've had exams, and then like you've taken those exams and you've shown me what you've comprehended and so forth. And as they were saying, I was only planning on taking on 1 student, and I had 3 interviews, and I couldn't choose between them, so I said you just start both of you. And I only meant to have them do their conditions, but they were so good that I would start noting them off with various stories and just develop your project. And it's because you're talented and they're committed and they're on time and they're but one thing that I want to say to you guys that are a bit younger is don't just think about this has to be your career path, because one huge thing you can learn from an internship is that this isn't what I want to do, or I want to do something similar or something different, because you're not meant to know what you want to do at this age. And I think, you know, like Carly, like, you've also learned what you like about it and what you don't like, and how you can tweak the future. And they're also getting, and this is unheard of in my field, they're getting acknowledgements in my papers because they really earned it. And that's something you can put on your CV and you can defend that and so forth. And it's really, really fun.
Do you have my CV? Curriculum vitae. Well, we talked in the car about kind of how workplace environments can be so different, where you end up like they might be really loud, or they might be really quiet, or kind of how important it is to find a work environment that suits you. How did you? Can you kind of describe what was your typical day like? How do things function here?
Well, since we were towards the end of our project, we were kind of working on the computers more. We kind of come in and then kind of get in our own zone, which works better for me. Like a lot of distractions in order, so just coming in, being able to listen to music and just work through the day is how I kind of work the best. But I know like friends of mine have done internships at like kindergarten and stuff, so it's kind of like whatever kind of personality type you are, that's where that's it. But for me, it was more just coming in and working on computers and that sort of thing.
They're very individualized.
Yeah.
Right, so we did a large portion of individualized work, and then we would also collaborate with each other at times to learn different things, such as the backpack prism, and we also did tissue culture together as well, and the staining process we also learned from Emma. So there were times when it was incredibly engaging, and times where we got to do our own different things. So it depends on the day, but generally for the most part, it was very structured.
What time did you come down on Mondays and Fridays?
We came in at around 4 p.m., or we tried to make it by 4 p.m., and then we leave around 6, so only 2 hours, but that's basically as soon as
Very interesting. You said, I mean, that you got a kind of key point that you wrote stuff down to either content or just something in general, just something that sticks out as you're gonna see the field.
Well, I learned that you really have to have a passion for the field of what you want to do, and Emma just loves doing this type of work. So, definitely finding my passion and finding where I really fit in is definitely key to my success. And I've seen that through Emma. So,
What's your least favorite part of your internship?
So can
Well, I think I've just seen a lot of times when you're like a lab technician or some sort of thing, you don't really get to pick and choose what you want to do. You're kind of like working for someone else always. So I see from other people that just putting in a few more years in school and just going for that PhD or the MD PhD really pays off when you're the overseer instead of the person that you're taking direction, which is completely fine if that's the kind of person you are. But I think it's just more beneficial if you're the one that gets to oversee the papers and like that.
Well, and some people like being told what to do.
Yeah.
Right? There's that personality thing with them.
And this is also like why it's good for them to have this kind of opportunity to turn maybe by soccer, because like that's something you can talk to with every team. They know they want to be the manager rather than the managed person. It's great.
Very nice, very nice.